Right, today we are going to look at structure of the cell. Remember we introduced anatomy last time. We said anatomy has three divisions: gross anatomy, microscopic anatomy, and developmental anatomy. Under microscopic anatomy, we said we have what we call cytology and we have what we call histology. So today we are going to start that aspect of microscopic anatomy, and specifically we are going to look at cytology. In this lecture, we are going to look at the principles of cell theory. We are going to look at the basic components of the cell. We are going to look at the structure of the cell as seen under light as well as electron microscopy. We're also going to look at cellular organelles and their major functions. I will take a small detour and also talk about the genetic makeup of the cell and how the chromosomes are organized. Let's begin with the first agenda there, which is principles of cell theory. In biology, you may have defined the cell as the basic unit of life. That is actually one of the principles of cell theory. We are calling it a theory and not a fact. The reason we are calling it a theory and not a fact is because maybe that statement is actually not so true. We don't have the evidence. And the reason it's arguable that maybe it's not so true scientifically is because if you're saying the cell is the smallest unit of life, then do you mean things which are smaller than the cell, like mitochondria, are dead? So that makes it a theory. It is difficult for us to conceptualize that organelles are dead, then now the cell is alive. So that's why, even though we say the cell is the basic unit of life, that is a theory. Second principle of cell theory states that any organism either has one or more cells. That is again true. I mean, you have multiple cells, billions and billions of cells in your body. There are some organisms that are unicellular, but that statement is still true, at least one cell. Organisms such as bacteria and protozoa, they are unicellular organisms. So as far as we are referring to those examples I've given you, that statement is true. How about viruses? Viruses are actually nucleic acids. You can have DNA viruses, you can have RNA viruses. Are they organisms? If we take this statement to be fully true, then it will eliminate viruses as being organisms. You people may not know, but there's some infectious agents, such as what we call prion, very infectious agents. You'll be told very, very infectious agents. But they're very small. They're actually proteins. They're very small, smaller than the cell. Are they organisms? So we don't know. And for that reason, we make this also a theory. The third theory of the cell is even more interesting. The third theory states that you cannot really form a cell from just de novo. For a cell to exist, it must have come from another cell through the process of cell division. And we know that's how we grow. I mean, a cell divides mitotically, then divide again, divide, you grow, you become the whole you. So that's true. But again, we question this because if it is possible that you actually cannot form a cell, that cells that exist can only exist because they come from pre-existing cells through the process of cell division, how then do you account for the origin of the very first cell? Definitely, it must have come from mechanisms which are not through cell division. Definitely. And that's why this number 3 is also a theory. So those are the principles of cell theory. What are the basic components of a cell? A cell has 3 basic components. The outermost cover is what we call the cell membrane. Please don't mention those things of CG, cell wall. The unit here is human anatomy, and so we are keeping it to human. We have cell membrane, we have what we call cytoplasm, and we have the nucleus. Those are the 3 basic components of a cell. If I ever ask you to use a label diagram to illustrate the components of a cell as seen under light microscope, then this is what you draw. Don't add anything in the cytoplasm if you're talking about light microscopy, unless you are a witch. Because in light microscopy, you cannot see beyond just saying this is the cytoplasm. You can't see the organelles. So these are the basic components of a cell. In light microscopy, it will look like this: cell membrane, cytoplasm, nucleus. These are white blood cells. These are epithelial cells, nucleus, cytoplasm, cell membrane. However, in electron microscopy, you can now maneuver your way out and talk about many things. The key point here is that in electron microscopy, we see details of the cytoplasm. More details of the cytoplasm are seen with electron microscopy. More importantly, we see that in the cytoplasm, there are organelles. So in electron microscopy, we're able to see the cellular organelles better, to the level that even if you're given just a photograph, it might be difficult for you to orient yourself. So that's I've given you this diagrammatic representation of what you expect to see in the cytoplasm. I want us to then discuss those 3 components of the cell. Let's start with the cell membrane. The cell membrane is described as being a phospholipid bilayer structure. Phospholipid bilayer structure. What does that mean? It means that you have 2 layers of phospholipids. A phospholipid is something with a phosphate group and a fatty acid chain. This is the phosphate group. Those rounded knobs, those are phosphate groups. And these strings are the fatty acid chains. So that's a phospholipid. So this is one layer of phospholipids, and this is the other layer of phospholipid. We have 2 layers of phospholipids, so it's a phospholipid bilayer. Now look at how the phospholipids are arranged. They are arranged in such a way that the fatty acid chains are sandwiched between the phosphate groups. The sandwiched parts of the fatty acids, the sandwiched parts of the cell membrane which contain fatty acids, are trying to be away from the water components, the regions with water, either within the cell or outside the cell. Therefore, this sandwiched part of the cell membrane is described as being hydrophobic. On the other hand, these regions of the cell membrane, the ones with phosphate groups, are actually in contact with the regions with water, either within the cell or outside the cell. And so these regions are described as being hydrophilic. They like water. Centrozone, hydrophobic, fears water. Apart from phospholipids, there are other components, biochemical components of the cell membrane. Importantly, we have proteins. As a matter of fact, even though we are saying the cell membrane is a phospholipid bilayer structure, which is the skeleton, as a matter of fact, the proteins on the cell membrane constitute the largest proportion of biomolecules in the cell membrane. 55% of the biomolecules on the cell membrane are actually proteins in nature. These proteins can be categorized in two ways. We can look at them anatomically, but we can also look at them functionally. Anatomically, we look at them this way. There are those proteins which are within the cell membrane, like these ones, or that, or that, or that. We call them integral proteins. The proteins which are actually incorporated within the cell membrane. We call them integral proteins. But there are also proteins which are on the sides of the cell membrane. They can be on the inner side like this, or outer side. You can imagine one being there or there. So those proteins are then called peripheral proteins. You can either have integral proteins or peripheral proteins. Those are anatomical classification of the cell membrane proteins. You can also have functional classification of the cell membrane proteins. Functionally, the proteins of the cell membrane fall under 5 or 6 classes. You can have a class which you are calling structural proteins. It means that they contribute the structural integrity of the cell membrane. You can have what we call receptor proteins. It means there are binding sites for things like hormones. They are receptors. You can have enzymatic proteins. It means they catalyze biochemical reactions. You can have antigenic proteins. It means they induce immunological responses. They are actually important for identity of the cell. When we say you are blood group A, it means there's a protein on your red blood cells called antigen A. When you're blood group B, it means there's a protein on the cell membrane called antigen B. So those antigens are a class of proteins. Then lastly, you can have what we call transport proteins. Transport proteins facilitate in movement of substances across the cell membrane. That's why we call them transport proteins. And there are two examples of transport proteins. You can have ion channels like this one. It's an ion channel, so ions go through it. But you can also have what we call carrier proteins. Carrier proteins and ion channels are examples of transport proteins. So physiologically, cell membrane proteins belong to those 5 or 6 categories of classes: structural, receptor, enzymatic, antigenic, or you can call that identity. Then you have ion channels and carrier proteins. Those last two are the ones we've called transport proteins. Apart from proteins, we also have other lipids which are on the cell membrane. In total, actually, lipids constitute about 42% of the biomolecules of the cell membrane. Examples of cell membrane lipids are actually phospholipids that we mentioned, as well as cholesterol. So cholesterol and phospholipids are examples of lipids within the cell membrane. The third category of lipids in the cell membrane are glycolipids. They are lipids embedded with some carbohydrates, glycolipids. Lastly, we have carbohydrates. Carbohydrates constitute about 3% of the cell membrane molecules. Carbohydrates. Nothing much about their role. Most of them are actually linked with proteins to constitute part of the antigenic functions. That is the structure of the cell membrane. So what are the functions of the cell membrane? We can say that the cell membrane protects the components of the cell. We can also say that the cell membrane is important for communication. One cell can communicate with another cell in the body. Such communications can be facilitated by their cell membranes, and especially if I give you examples, a cell can produce a hormone, and that hormone goes to another cell. So that hormone will bind on its receptor on the cell membrane of that other cell, the recipient cell. So for that reason, cell membrane is facilitating communication. Another example I can give you is in neurons, where neurons transmit electrical impulses, but those electrical impulses are actually being transmitted along the cell membrane of the axon. So that is examples of intercellular communications. The cell membrane is also important for transporting substances across it, either into the cell or out of the cell. For large molecules, movement into the cell is what you'll call endocytosis, and movement out of the cell will be called exocytosis. That happens for large molecules like bacteria. Now for endocytosis, if that large particle is fluid, then the correct term to use is pinocytosis. If that large substance is solid, then the correct term to use is phagocytosis. Phagocytosis and pinocytosis are examples of endocytosis. Then exocytosis does not matter the substance of what is being expelled. As long as the cell is expelling something large, you call it exocytosis. For small molecules, you don't use those terms endocytosis and exocytosis, because small molecules, things like sodium ions, potassium ions, will now move through different mechanisms, and they tend to move in form of a rush. So when small molecules enter, you call that influx. And when small molecules come out of the cell, you call that efflux. So you can have sodium influx, sodium efflux, potassium influx, potassium efflux. There are better terminologies that you'll use in physiology to describe movement of small molecules. So you'll talk about things like passive transport and active transport. The other role of the cell membrane is in cell recognition or identity. And this is because the cell membrane contains antigens on its structure, that are responsible for identification of cells. I've given you examples of when we say you are blood group A, it means your red blood cells have an antigen called antigen A. If you are blood group B, it means your red blood cells have an antigen called antigen B. If you are blood group AB, it means you have antigen A and antigen B in your red blood cells. If you are blood group O, it means you have none of those antigens. It doesn't mean you have antigen O. We don't have antigen O. It just means 0. If you are rhesus positive, it means you have the antigen for rhesus factor. That antigen is called antigen D, D for dog. So if you have antigen D, then we say you are rhesus positive. If you don't have antigen D, then you are rhesus negative. Other examples I can give you, you heard about people with HIV, then you hear we talk about CD4, CD8. That CD4 is actually an antigen on the cell membrane. It stands for what we call cluster of differentiation, CD. That is different from the CD you might be having in mind at the moment, but let's just go on. Right. I want us to talk about something else before we talk about the nucleus. So we've talked about the cell membrane. I want us to talk about the second component of the cell, which is the cytoplasm, before we talk about the nucleus. So let me just get that right. Okay. Let's talk about the cytoplasm. The cytoplasm consists of three things. We have the cytosol, we have cytoskeleton, and we have cellular organelles. Let's look at each of these three. We begin with the cytosol. So the cytosol refers to the fluid components of the cell. Simply, it's just water. The water within the cytoplasm is what we call cytosol. Why? What is the importance of this water? In as much as it holds the organelles, it also forms a medium within which biochemical reactions can occur within the cell. And that is its importance. It's a medium within which chemical reactions can occur. The second component of the cytoplasm is the cytoskeleton. Cytoskeleton refers to a network of protein filaments within the cytoplasm. There are three types of cytoskeleton. We have what we call microtubules. Microtubules, as the name suggests, are tubular, but they are small, microtubules. In as much as they are small, they are the largest component of cytoskeleton. They have a role in cell division because they form part of the centrioles. They form part of the centrioles, and so they have a role in cell division. They're the ones that help to pull aside the parts of the cell during cell division. And that means that if you're going to manage things like cancer, you can actually target drugs which inhibit microtubules. They will inhibit cell division, so you may arrest cancer growth. But of course, you'll also expect some side effects. It may not affect just cancer cells, it can also affect any other cell in the body that is dividing very fast. That's why when we give people with cancer some drugs that target cancer, they also tend to be anemic because we also affect blood cells which divide very fast. Maybe they lose hair because hair, again, the cells of hair grow very fast. Right, apart from being important in cell division, microtubules also form part of cilia. You remember cilia is found in a respiratory tree, even in fallopian tube. Cilia is usually motile. The structure of cilia contain microtubules, which is a motile structure. Lastly, microtubules are also found within the flagellum of the sperm. So they're the ones that help the sperm to swim from the site where they're deposited to the site of fertilization, hopefully. Right, those are microtubules. The second type of cytoskeleton is what we call microfilaments. These are the smallest of the cytoskeleton. Microfilaments are also called actin filaments. Actin filaments are important in muscle contraction. Usually, you have what you call actin and myosin. You have actin and myosin. So actin work together with myosin to help in muscle contraction. The other role of microfilaments, apart from being important in muscle contraction, is that microfilaments also form part of what we call microvilli. Microvilli are extensions of the cell membrane, usually on the apical surface, that facilitate transport of molecules because it increases the surface area of the cell membrane on the absorptive side. So the integrity of microfilaments are maintained, the integrity of microvilli is actually formed by microfilaments, the actin filaments. The third type of cytoskeleton is this one, which we call the intermediate filaments. As the name suggests, their size is in the middle between microtubules and microfilaments. Intermediate filaments are important in providing structural support to the tissues they are in. However, what is unique about intermediate filaments is that they are unique depending on the tissues they are found in. For example, in epithelial tissues, we call them keratin. In muscle tissue, we call them desmin. I've given you two examples. For epithelial tissues, call them keratin. I'm sure you've heard of keratin before, but maybe you've never heard of desmin. Desmin is what we call intermediate filaments in muscle. Now, why am I insisting on that point? I'm insisting on that point because there's something clinical I want you to know about it. If intermediate filaments are unique for different tissues, then it means that we can utilize that information clinically, especially in diagnosis of cancer. Let's say someone has cancer in the brain. You don't know whether it's primarily from the brain or it's cancer that was somewhere that spread to the brain. So to distinguish those two, when you biopsy that cancer tissue, you may want to then find out which type of intermediate filaments those cells do have. If you realize that they have keratin, then you definitely know that that cancer is of epithelial origin. It is not a tumor in the brain primarily. It came from somewhere, then went to the brain. So we use intermediate filaments in tumor mapping or as tumor antigens, so to speak. There are molecules we can use to identify tumors, tumor antigens or tumor mapping. Right, so those are the cytoskeleton. We've talked about three types of cytoskeleton: microtubules, microfilaments, and intermediate filaments. The third component of the cytoskeleton are the cellular organelles. So, cellular organelles are structural units within the cytoplasm that perform specific functions of the cell. There are many organelles. You have mitochondria, ribosomes, endoplasmic reticulum, which can either be smooth and rough, Golgi apparatus, secretory vesicles, lysosomes, centrioles, and I want you to add peroxisomes on that list. There are very many. Now, I find it a bit a waste of time to take you through functions of cellular organelles. This is what you did in high school. But because I still want you to know it, and I hate repeating it, I want to give you time to discuss it amongst yourselves. So, I'm going to divide you in small groups, and during that division in small groups, I want to, okay, I can see you people are 18 in total. I was expecting 20 something, but it's okay. So during that time, I'll also take the class attendance. So you are 18. I'll divide you into 3 groups. That means you'll be 6 per group. Okay, so I think you'll get a pop-up to tell you where you belong. What I want you to do is to state the key functions of each of these cellular organelles, and you add their peroxisomes as well. So join your groups, discuss. You're doing this in 10 minutes. So welcome back. I believe you discussed the functions of the different cellular organelles. So now we are done with the second component of the cell, which is the cytoplasm. Now I'll take you back to the third component of the cell, which is the nucleus. Remember, we said the cell has three basic components: cell membrane, cytoplasm, and the nucleus. We've done the first two, so let's do the last one. The nucleus is an important part of the cell. Most cells will have just one nucleus, but there are other cells with multiple nuclei. For example, skeletal muscle cells will have multiple nuclei. Cardiac muscle cells may also have two nuclei. There are some cells which don't have a nucleus. A good example of a cell that lacks a nucleus is the red blood cell. The nucleus is covered by a membrane. You can call it the nuclear membrane. The other name given to the nuclear membrane is a nuclear envelope. This envelope has some holes within it. These holes are known as nuclear pores. Through these holes, substances can move into and out of the cell. Into and out of the nucleus. The fluid within the nucleus is called nucleoplasm. Nucleoplasm. The fluid medium within the nucleus is called nucleoplasm. The fluid medium outside the nucleus is called cytoplasm, as we've already seen. So, nuclear pores allow communication or movement of substances between the cytoplasm and the nucleoplasm. What are the functions of the nucleus? The nucleus controls the activities of the cell. The nucleus is also a site for synthesis of ribosomes. You'll be told that ribosomes have two components. Or when you're looking at functions of ribosomes, some of you may have discussed that in your breakout sessions. Ribosomes have two components. They have an RNA component and protein component. The RNA component of ribosomes are synthesized within the nucleus. The specific region within the nucleus that synthesizes ribosomes is known as the nucleolus. So nucleolus is for synthesis of ribosomal RNA. Ribosomes have an RNA component as well as protein component. So the RNA component are synthesized within the nucleolus. The third major function of the nucleus is that it stores and protects the genetic material of the cell. Not all genetic materials are contained within the nucleoplasm. Some genetic materials of the cell are actually within the mitochondria. So mitochondria usually has its own DNA, mitochondrial DNA. But the nuclear DNA is the major genetic material. The nucleus protects and stores genetic material. So I want us to take a small detour and talk about that genetic material of the cell. The genetic material of the cell, we've said, is what we call DNA, which stands for deoxyribonucleic acid. Usually, the genetic material exists in form of what we call chromatin material. This is what we call chromatin material. The chromatin material refers to DNA that has been combined with proteins. The major proteins of the DNA are known as histone proteins. Histone, histone proteins. These histone proteins are the major proteins that are combined with the DNA to constitute the chromatin material. There are two forms of chromatin material. There is this chromatin material that appear lighter in this region here. This type of chromatin material is known as euchromatin. So euchromatin is this type of chromatin material. It is the dispersed chromatin material, this one. Usually it appears lighter, and the term we use for it is that it is electron-lucent. It is electron-lucent because it is dispersed chromatin material. It's like it has opened up itself. Such type of chromatin material is the one that is active in transcription. You'll remember that transcription is a process of forming messenger RNA from a DNA template, and that is an important step during protein synthesis. So euchromatin will be more in a cell that is active in protein synthesis. The opposite of euchromatin is heterochromatin, which is this one. Heterochromatin is dense, as you can see here. So the term we use is that it's electron dense. It is electron dense because heterochromatin is condensed. Chromatin that has condensed itself. Such type of chromatin is not easily available for transcription. So it is not active transcriptionally. Okay, there's some people asking questions. Okay, so for the proteins, I said the histone proteins are the proteins which combine with DNA to form chromatin. I'm going to mention about them again shortly. Right. Now I want us to see how the DNA of the cell, how the DNA of the nucleus is organized. In high school, you must have talked about DNA and RNA. And one of those differences you talked about is that DNA is double-stranded and RNA is single-stranded. That double-stranded molecule is the lowest level of organization of the DNA. That the DNA is a double-stranded molecule. We call that the lowest level of DNA organization, the double-stranded molecule. You may still remember what these codes mean. Remember, these are the bases, A, T, G, C, how they pair with each other. That is not our interest in anatomy. Whether you remember or you don't remember, in anatomy, I don't think I care much there. But just know that the DNA is double-stranded. So there's one strand and the other strand there. And usually the bases pair in a particular way. Now I want you to follow this double-stranded molecule and notice that at some level, the double strand molecule twists on itself. So for example, if you see my fingers, this is double strand molecule, then the two strands twist on themselves like this. That twisted DNA molecule forms the second level of organization of the DNA, which we call the DNA helix. So the helix structure is the second level of DNA organization. Now, what happens to the helix? You can imagine the helix to be something like a rope. Now, which type of rope? This rope that you make from sisal. So they're usually twisted amongst themselves like this. Imagine that being the helix. Then now that rope goes around some proteins. The proteins associated with DNA, I told you, are called histone proteins. So the helix structure usually go around histone proteins twice in this manner. The helix structure goes around the histone protein twice. A histone protein that is surrounded by the helix structure twice constitute the third level of DNA organization, which we call the nucleosome. So the nucleosome is a third level of DNA organization. Histone proteins that is surrounded by the helix structure twice. Several nucleosomes compact or group together to form chromatin material. And that is the form in which the genetic material will exist in our nuclei, the chromatin material. Now, most of the time, the story will end there. However, if this cell ever needs to divide, if a cell needs to divide, we will see other forms of organization of the DNA that we do not necessarily see if there is no cell division. Before I mention that, I want to ask you a question. Let's assume that we can count the genetic material. Let's assume that there are 20 DNAs, so to speak. I am a cell, I want to divide. If I just divide, remember I'll cease to exist, then I'll give rise to two daughter cells. Those two daughter cells will have to share my genetic material. So one will have 10 and another will have 10. If those daughter cells ever want to divide again, they will have to share their genetic material. So the daughter cells will have 5, 5. Those are now my granddaughters. Now you realize that if division occurs in such a manner, then at some point the genetic material will cease to exist. You will be not a human being. So division does not happen in that manner. Now, my question to you is, what should a cell that needs to divide do to ensure that the daughter cells will have the same genetic material as the parent cell? That's my question to you. What should I do to ensure that my daughter cells will also have 20 instead of half what I have? Anyone can unmute themselves and answer. Are you people in class?
Yes.
Okay, then try answering.
replicates itself.
Very good. But let me correct what you said. The cell will replicate its DNA, not itself. It will replicate its DNA, which means that if I had 20 DNA, before I divide, I will make another copy of my DNA. So I will have 20 and 20. I will have 40. I will have two sets of the same genetic material. Then now I divide. When I divide, one cell will have 20, another will have 20, the same genetic material. It is the same thing that also happened to cells. When a cell needs to divide, it will replicate its genetic material so that it will have two sets of the same genetic material. These two sets of the same genetic material then condenses. So when they condense, they become visible chromosomes. You can see them. They become visible chromosomes. Like this, or better still, like this. This is a chromosome. What I want you to realize that even though this is a chromosome, it will be having two sets of the same genetic material. One set is called a chromatid. The other set is also called a chromatid. So two chromatids constitute one chromosome. Therefore, the fifth level of DNA organization is the chromatid. And the sixth level of DNA organization is the chromosome. The chromosome is the highest level of DNA organization. Having said that, I will then ask you some questions shortly. But let's look at this first. This is how chromosomes in humans appear. We have a total of 46 chromosomes in humans. But these 46 chromosomes are actually 23 pairs of chromosomes. One pair came from your mother. Okay, my internet is a bit unstable. Just a second. Okay, I think it's better now. So we have a total of 46 chromosomes, which is actually 23 pairs of chromosomes. One pair came from your mother, and another pair came from your father. So yes, there are two sets. One set is of maternal origin, 23. Another set is of paternal origin, 23. So a set of chromosomes is a set of 23 chromosomes. 23 chromosomes constitute one set. When they're in a cell, we still call them chromosome 1 up to 23, but they're in pairs. Having said that, when a cell has 46 chromosomes, we describe that cell as being diploid. When a cell has 23 chromosomes, we describe that cell as being haploid. So, diploidy is when you have two sets of the chromosomes. Haploidy is when you have one set of the chromosomes. That means that we can also have triploidy and even quadriploidy. Triploidy is when you have 69, and quadriploidy is when you have 92. chromosomes. I hope you've understood those terms. I'm insisting on them because I'm going to introduce another term that I'll want to distinguish the two. But let's see. How do we name the chromosomes? I've told you there are 23 pairs, but we name them like this. The first 22 pairs up to there is what we call autosomes. That's why you hear some genetic disorders being autosomal dominant, autosomal recessive. It means that they affect those chromosomes between 1 to 22. The 23rd pair of chromosome is what we call the sex chromosomes. Sex chromosomes are of two types. You can have the X chromosome. You can also have the Y chromosome. Those are the two types of sex chromosomes. So disorders that affect the 23rd pair of chromosome are sex-linked disorders. They can either be X-linked or Y-linked, but they are sex-linked disorders. They are not autosomal disorders. Right, so this is the 23rd pair of chromosome. Having said that, let me now ask you a question. So I want you to look at these things. Whatever you're looking at right now is what we call karyotype. A karyotype is the graphic representation of chromosomes within the nucleus of an individual or within the nucleus of a cell from an individual. Karyotype. I want you to look at the left karyotype as well as the right karyotype and tell me whether they are normal or abnormal. We are focusing on the number of chromosomes, not on the structure of the chromosomes. I haven't taught you anything about chromosomal structure, and I'm not going to, so let's leave that aside. We are focusing on chromosomal number. The one on the left, is it normal or abnormal? The one on the right, is it normal or abnormal? So anyone, just unmute your mic and tell me something.
On the left, it's abnormal.
Okay, so what makes you think that the left one is abnormal?
Because the 23rd chromosome that consists of the X and Y, the Y is not there.
Okay, but let's focus on the numbers. Remember, we said that we have 23 pairs of chromosomes. That is 22, then this is the 23rd pair. How many chromosomes do we have on the 23rd pair here? We still have 2.
Okay.
So, is it normal or abnormal? Anyone?
It is normal.
It is normal. Both of these are normal human karyotypes. Now, how do we write this one? This person has 46 chromosomes in total. So normal. The 23rd pair of chromosomes consists of 2 X's. So we write it as this, 46XX. 46XX is a normal karyotype of human female. There are 46 chromosomes. The 23rd pair consists of 2 X chromosomes. The one on the right is still 46. The 23rd pair, there is 1 X and 1 Y. This is the normal karyotype of human male. 46XY means normal male human. 46XX means normal female human. Right, let me give you something more. Again, you are presented with two karyotypes. There's one on the left and there is one on the right. Normal or abnormal? Let's start with the left one. Anyone?
I think the left one is normal.
Okay, you think the left one is normal. How about the right one?
I don't think it's normal.
Okay, so where is the abnormality, Adelaide?
It's because it has, it has both.
Yes, both?
Both normality for female and male, which actually,
Sorry?
it has both normal karyotype of like both male and female. So I'm not even sure. I'm kind of confused. So I'm not even sure if
So
it's
Adelida,
okay or it's not okay.
remember the main aim of this session is to talk about the number of chromosomes. Number. So, is the number on the right image normal or abnormal?
Abnormal.
Okay, what makes it abnormal?
Because it has one extra.
There is one extra chromosome. On
Yeah.
which pair?
On the female. X.
On the 23rd pair.
Yeah.
There is one extra chromosome on the 23rd pair. I want you to look at the left one again. You'll also notice that on the 21st pair,
Oh, yeah.
there's an extra one. So these two are numerical chromosomal disorders.
Okay.
When you have a numerical chromosomal disorder, you call it aneuploidy. Aneuploidy is numerical chromosomal disorder. Now let's look at the left aneuploidy. The left one, you have one extra chromosome in the 21st pair. That means that this individual will have a total of how many chromosomes? Anyone?
47.
Very good. When you have 3 chromosomes.
7.
All right. Wengine bando zao zinafika 5 minutes later, that's okay. When you have 3 chromosomes in one particular pair, we call that trisomy. Don't confuse trisomy with triploidy. When someone is trisomic, we want to know which chromosomal pair is actually trisomic. So in the first one, we can see that it's affecting chromosome number 21. So we call this trisomy 21. Someone with trisomy 21 is someone with Down's syndrome. Now, let's revisit that. You've agreed with me that when you are trisomic, you have 47 chromosomes in total. We are making an assumption that the trisomic one is only affecting one chromosomal pair. Now, let's visit this other one. This one, we've called it trisomy 21. How do you then call this one? Anyone? Hey, just talk. I've asked you a very simple question.
Trisomy, I think it's trisomy 23.
Very good. Thank you, Tabi. Trisomy 23. However, we don't call it trisomy 23, but you're right. It's a trisomy 23. Now, the reason why we don't call it trisomy 23 is because we've agreed that for the 23rd pair of chromosomes, there are two options. You can either have X or Y chromosome. If you tell me that this person is having trisomy 23 disorder, then I keep on wondering among four options. Is it trisomy 23 because there are 3 X chromosomes? That's one option. Or there are 2 X chromosomes and 1 Y, like we have here, that's second option. Or there is 1 X and 2 Y, that's third option. Or there are 3 Y chromosomes, if possible, fourth option. So if you write in the diagnosis that this is trisomy 23, we will still need to know more about that. For that reason, we don't classify this person, or rather we don't write it as trisomy 23, in as much as that is still true. So we have to use a different coding style to write this karyotype. You see, for this one, it's very simple, trisomy 21. It means 3 chromosomes on the 21st pair. But if you say trisomy 23, it will mean 3 chromosomes on the 23rd pair, yes, but I still remain with some questions in my mind about the pattern. So now we've agreed that if you have trisomy, you have a total of 47. It therefore means that we can use the letter 47 to mean trisomy. And we can use the letter 45 to mean monosomy. Monosomy will then mean that there's only 1 chromosome in a particular pair. So, having said that, this one is written as 47XXY. It means you have a trisomy on the 23rd pair of chromosomes, and this is the pattern on the 23rd pair of chromosomes. XXY. Someone with 47XXY is someone with Klinefelter's syndrome. Right.
What syndrome?
The spelling is there. Can you see it? Klinefelter syndrome. Right. I want us to think through something. Look at the image on the left. Also look at the image on the right. Try to tell me the gender of these individuals. Let's start with the left one. Male, female, or both? Okay, let's drop the option of both. It's either male or female. So the left one, male or female?
Male.
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 Good, good, good. How about the right one, male or female? I'll give you the option of both here.
Female.
Female.
Okay. Both of them are male. This one is male. Why do you think I say so? Anyone?
Because of the Y chromosome.
Very good, Roslyn. Now the key is, when you're talking about gender determination at a genetic level, the genetic level of gender determination does not depend on the X chromosome. It depends on the presence of the Y chromosome. Genetically, if there is a Y chromosome, it means genetic male. And if there is no Y chromosome, it means genetic female. Genetically, that is female if there is no Y chromosome. And if there's Y chromosome, that's genetically male. It does not matter whether you have X chromosome or not. As long as there is no Y, you are female genetically. And if there is Y, you are genetically male, even if you have 10 extra X chromosomes. So this person is genetically male. This is genetically male. I am using the term genetically because when we have that lecture on sex determination, we will see that there are 3 types of sex. There's genetic sex, which either says whether you are X or XY, where XY will mean then male and XX will then mean female because there is no Y. But there's also the gonadosex, which means do you have a testis or an ovary? Then there is the anatomical sex. The anatomical sex is what you're familiar with. You look at the external genitalia, is there a penis? Male. Is there a vagina? Female. So I'm using the level of genetic sex. So genetically, this person is male. Phenotypically, there could be something ambiguous about it, and we'll talk about that later. Right. I want you to look at this other one. So someone look at it and tell me, how do you write this karyotype? Bear in mind that this one, we call it trisomy 21. And I gave you reasons why. This one, we call it 47XXY. And I told you why we cannot call it trisomy 23. So bear in mind this naming system and this naming system. And when they both apply, then give me how we'll call this one. So someone can try, give me a genetic code for this one.
You had mentioned monosomy 21. I think it's this one.
Okay, so this is monosomy what?
21.
Okay, look at it again. Is it really a monosomy 20? It will be?
Monosomy 23.
Monosomy 23. Okay, I agree with you partially. You remember the concept of the 23rd chromosome being either X or Y?
Monosomy 23.
I agree with you that it's a monosomy 23, but remember I told you that 23rd pair has X and Y. Same concept for trisomy 23, we don't call it trisomy 23, in as much as that's still true. So even here, we don't call it monosomy 23, even if that's true. We like to use the other naming system. So we call it 45X0. Don't call it monosomy 23. Because if you are monosomy 23, it means that there is one chromosome on the 23rd pair. That one chromosome can either be X chromosome or Y chromosome. So we want to specify. So we write it as 45 to mean monosomy. Then we write the chromosome that is present. This 0, you can leave it or put it there. I like putting it there to mean that it wasn't an error to omit the other one. It's actually the other one is not present. We don't know which one is absent. It's either the X chromosome that is absent or it's the Y chromosome that's absent. We don't know. Either way, this is what we call Turner's syndrome. So someone Turner syndrome will have 45X0. Tell me, they have only one X chromosome. Will they be female or will they be male? Someone Turner syndrome.
Female.
Okay.
Female.
Yes, I agree with you. They'll be female, and you've understood why they'll be female. This other one has 3 X chromosome on the 23rd pair. How do you want to give this the genetic code? Anyone?
47.
47XXX.
Very good. So this is 47 triple X. That's what we call triple X syndrome. How about this one? Anyone?
Trisomy 18.
Very good. Someone with trisomy 18 is someone with what we call Edwards syndrome. Lastly, this one. How do you want to call that?
Trisomy 13.
Trisomy 13. Good.
Trisomy 13.
Trisomy 13 is someone with Patau syndrome. All right, ladies and gentlemen, we have gone through the 3 parts of the cell membrane, the cytoplasm, and the nucleus. In the nucleus, we've talked about the fact that it has a membrane. We've talked about the functions, and we've said that one of those functions is that it contains and protects genetic material. We've also taken a detour and talked about the genetic material of the nucleus, how it's organized into chromosomes from the simplest double-stranded molecule. Then we've seen the chromosomal pattern, at least the numerical one, in humans, and we've given examples of common numerical chromosomal disorders. So we are now done with the lecture that we are supposed to have today. I believe you're able to answer the questions that are projected. Good. So we are done with cytology. I told you earlier that you need to read on cell cycle and cell division. So take time and read that one, cell cycle and cell division. You will need that information sometime later, maybe in the course of next week. Tomorrow, let me check if we are meeting. Just a second. Okay. We aren't meeting tomorrow. We are meeting next on Monday. So our class on Monday, we are going to start on basic histology. We'll be looking at epithelial tissues on Monday. Thank you very much. Unless there's a question, I will now end the session. Is there a question? No question. Thank you very much. Have a good afternoon.
You too.
Two.